Your last questioner, Jordan Clark, is going to now talk about the protective capabilities of non-neutralizing antibodies elicited SARS-CoV-2 vaccination. And so Jordan works at the, I'm going to say, the ICN School of Medicine at Mount Sinai. And sorry if I pronounced that incorrectly or suboptimally. And yeah, Jordan, we're really interested to hear you give this presentation. Thank you.
Thank you very much for having me. It's great to speak to you. Thank you very much for inviting me. So I just gave this talk at the Options Conference in Belfast. So if you attended that, I'm sorry, you might have already seen this. So yeah, my talk is pretty much, as you said, focusing on non-neutralizing antibodies, which are elicited in response to vaccination. And we really wanted to look into these to see if they were protective or not. Okay, I won't go into much detail on this. I think Ed already covered this, and I mean, we're 2.5 years into the pandemic, so we all know what SARS-CoV-2 is. Suffice to say that spike is the main glycoprotein. It's present on the outside of the virion, and it, of course, mediates cell entry via interaction with ACE2, and it is the major target for neutralizing antibodies. It can be, it's composed of homotrimers, which can be split into two different domains, S1, which contains the N-terminal domain and the receptor binding domain, and S2, which contains the fusion peptide and the transmembrane domain. So in a previous study, which was conducted by the lab before I joined in 2021, they wanted to investigate the antibody response following vaccination. And to do this, they took 3 people who had received the Pfizer vaccination. They had PBMCs taken, plasmablasts were isolated, and antibody sequences were cloned by one of our collaborators, Ali El-Abedi. So from these 3 people, they managed to clone 42 anti-spike monoclonal antibodies. And through ELISA, using different subsections of spike as a substrate, it was found that 12 of these bound RBD, 10 bound NTD, 17 bound S2, and 3 bound unknown epitopes. But the major finding of this publication was that only 7 of these antibodies were neutralizing. And it seemed that the majority of the antibody response in these 3 participants was for non-neutralizing antibodies. So we wanted to build on this and look into whether or not these non-neutralizing antibodies are protective. So to do this, I took these 42 antibodies, and the first thing I did was look at the binding of these antibodies to different spike variants. So you can see down here on our x-axis, we have various different spike variants, including Omicron just here at the end, BA1. And on the y-axis, I'm showing the minimum binding concentration. So on these graphs, if the minimum binding concentration increases, it means that there is a loss of binding. So wherever you see more intense color here, that means that there is a decrease in binding against these spike variants. And I've noted these 7 neutralizing antibodies with little red ends, and then the rest of these are non-neutralizing. So you can see, looking to the RBD binding antibodies, only a couple of them really seem to lose any kind of binding against the variants. And indeed, the two neutralizing antibodies maintained binding, which was perhaps a little bit surprising, since several of these variants, have a lot of mutations within their receptor binding domain. But it looks as if the majority of the non-neutralizing antibodies here retain binding. Looking to NTD, where we had a few more neutralizing antibodies, you can see that 3 of them have lost binding to a couple of these variants. However, the majority of the non-neutralizing antibodies maintain good binding. The same for S2, you can see there's 2 non-neutralizers down here that lost a little bit of binding to P1, P3, and A231. The majority of them retain binding. For unknown binders, 2 of them lost a little bit of binding, but one of them is essentially unaffected. So it seems like the majority of these non-neutralizing antibodies are still able to bind to these different spike variants. So to determine whether or not they're protective, we used a mouse challenge model. So we treated mice with 10 milligrams per kilogram of antibody intraperitoneally, and 2 days later infected with a 5 times LD50 dose of the ME10 mouse-adapted SARS-CoV-2. So you can see the weight loss and survival curves here, and you can see for for RBD, the 2 neutralizing antibodies were 100% protective, but so too were several of the non-neutralizing antibodies. Similarly, for NTD, all of the neutralizers were protective, but so was 1 non-neutralizing antibody. For S2 binders, you can see 5 of them there are protective, and indeed so is 1 of our unknown epitopes. So of the 42 monoclonal antibodies that I looked at, 19 were found to be protective, and of this, 12 were non-neutralizing. So it seems as if these non-neutralizing antibodies can mediate protection against SARS-CoV-2, which is good news. So we repeated this challenge experiment using a 1 times LD50 dose instead, and then looked at lung to viral titers at 3 days post-infection and 5 days post-infection. For 3 days post-infection, you can see that 3 of the neutralizing antibodies reduced lung viral titers below the limit of detection, but the entire subset of 19 antibodies reduced lung viral titers compared to the negative control, with the exception of this one here. At 5 days post-infection, you can see that the majority of the antibodies have promoted the clearance of virus, and all of them are lower than the negative control, which was a flu antibody. So it looks as if these antibodies can reduce lung viral loads and promote protection. So how might they do this? There's 2 mechanisms which I'll cover here, ADCC and ADCP. So you can see we have SARS-CoV-2 infecting an epithelial cell, and this results in a lot of spike localization to the cell surface. This can then be bound by antibodies, which in turn can be bound via their FC regions by different effector cells. So here I'm showing an NK cell. Its FC gamma receptor can bind the FC region, and this results in the production of, or the release of granzyme B and perforin, which kills the infected cell. That's antibody-dependent cellular cytotoxicity, or ADCC. Similarly, these can be bound by phagocytes using their FC gamma receptors, and this results in the phagocytosis of the infected cell. That's antibody-dependent cellular phagocytosis. 2 very verbose words, or verbose terms. We also have, these antibodies can also be bound by complement. However, I'm not going to cover that in this talk. So we can assay this using a commercially available luciferase reporter assay. To do this, we infect Vero E6 cells with the recombinant Newcastle's disease virus, which expresses hexapro spike. This can be done at BSL2 or CL2 if you're in the UK. We infect them with an MOI of 2, and then 48 hours later, we add our antibody dilutions, and then these commercially available effector cells. So these express different FC gamma receptors, either 3 for the case of ADCC or 2 for the case of ADCP, and that's connected to a luciferase expression gene pathway. So 6 hours after the addition of these, we then read luminescence, and we can tell whether or not these antibodies mediate ADCC or ADCP. So you can see, this is our readout here, you can see that the majority of the antibodies have ADCC activity. A few of them don't have any, and a few of them are a little bit variable, but it looks as if the majority of them do mediate ADCC. And indeed, the neutralizing antibodies here seem to have the most activity in this reporter assay. The ADCP assay isn't quite as reliable. So what I'm showing here is 1 representative experiment of 2 repeats. And as you can see, less of them seem to exhibit ADCP activity. But we think that this is likely the mechanism through which these antibodies are eliciting protection. For the ones that don't seem to have any or much activity, they could be mediating protection via interaction with complement. But I haven't quite gotten around to doing any complement assays, so that'll be one of the next things I'm going to look at. So to investigate this a little bit further, we utilized 2 well-characterized antibody mutants. This is the LALA mutant, which is leucine to arginine mutation 2 leucine to arginine mutations sites 234 and 235 within the FC region. This leads to a reduction in FC effector binding, whereas we also have the LALA PG, which is the same as LALA, but it has an additional mutation, and this results in the abolishment of any effector binding. So we can use these mutant antibodies to reduce effector binding and then determine whether or not the protection elicited by the antibodies is due to these functions. So this is just me repeating my ADCC and ADCP assay with these mutant antibodies. And as you can see, the wild type have activity, whereas the LALA and LALA PG do not. So this just proves that this is working. And we predicted that these mutations would reduce the protective capabilities of these antibodies. So we repeated our mouse challenge study using wild type and LALA PG versions of the 7 most protective antibodies, 6 of which were non-neutralizers, 1 of which was a neutralizer. And you can see here for this NTD binding non-neutralizer that in the presence of the LALA and LALA PG mutations, we lose protection, which is perhaps to be expected. For this RBD binder, you can see that the LALA loses a little bit of protection and the LALA PG more so. And for this non-neutralizer, you can see the LALA is still protective in this case. We had one mouse from the wild type group die. That's why it's appearing down here. The LALA is still protective, whereas the LALA PG no longer is. So since the LALA is just a kind of partial silencing, you might expect there to be some effector binding. So it could be that there's some residual binding going on here, which is what leads to protection in this group. So these results were kind of to be expected. However, the other antibodies we tested, we kind of saw something surprising. So we have one neutralizer in here, which I included as a kind of control. And you can see that in LALA and LALA PG backbones, it is still protective. You may expect that since it is a neutralizer, it can still neutralize virus. However, these other three antibodies, one S2 binder, one RBD binder, and one unknown epitope binder, even in the presence of the LALA and LALA PG mutations, they are still protective. So these antibodies cannot neutralize virus, and they no longer how they can still be conferring protection in these mice. So this is kind of the next big question that we're going to try and answer. So just to conclude, I've shown you that non-neutralizing antibodies are produced in response to vaccination, more so in these subjects than neutralizing antibodies. And these non-neutralizers retain binding to these different SARS-CoV-2 variants of concern. And of the 43 monoclonals that I assayed, 19 were shown to have protection in the lethal mouse challenge model, and 12 of these were neutralizers. All 19 reduced lung viral titers, resulting in clearance of virus by day 5 for the majority of the antibodies, and several of them, if not the majority of them, exhibited ADCC activity. However, when we cloned these into effector backbones, 3 of the 6 most protective non-neutralizers were still protective, and we don't understand how these non-neutralizers, which don't have effector functions, can be protective. So for future studies, I want to look into the complement pathway and see if some of these antibodies are working through that. And also, we need to repeat our LALA and LALA PG challenge using humanized Fc gamma receptor mice. We have a collaborator in Colombia who can do this. Unfortunately, those mice are on a K18 backbone, and all of the work I've done so far is using mouse-adapted virus and BALB/Cs. So we're going to have to repeat it in K18s to make sure we still get the same phenotype, and then we're going to have to repeat it in humanized Fc gamma receptor mice. So unfortunately, I have a fair amount of work to do. That just leaves me to thank everyone at the Kramer Lab who's helped. Particularly Fatima, who's I inherited this project from, Irene, who helped with the mouse experiments, all of our serological core members who helped with ELISAs, Viviana Simon, Goran Bajic, Wayna Sun, and at St. Louis, Ali Alabedi. And thank you very much for listening. I'm happy to take any questions.